this is the functional unit of the kidney here. And, if you look at the kidney under a microscope, it's arranged in these little hexagonal shapes. So this is histology. And by studying it, we can learn the physiology of how the liver operates. When we say lobules, it's roughly that six-sided shape. There's a hole in the middle. And so what I want to remind you about the liver, it has a dual blood supply in a single venous drainage. And we can see that in the lobule. So look for a dual blood supply. In a single venous drainage. Now go back and look at your notes, but the dual blood supply was the portal vein and proper hepatic artery. And the single venous drainages are hepatic veins. Now, so we can see that in the lobule structure. What we're going to see as we look further is blood will flow toward that hole in the middle, which is the central vein. That's your drain. I call it the shower drain. That is the single venous drainage. And but as the liver produces bile, those flow the opposite way to these corners. And so, really, on a real histology slide, the only thing I'll expect you to identify is the central vein and the portal triad, which are three structures on each corner of the hexagon-shaped lobule. So pointing to those two things, it's not hard to tell which one's in the center, right? That's the central vein right there. Portal triad on every corner, although this is the only corner where I see structures. The portal triad, I usually have to use pictures to teach what's in the portal triad. Because triad implies how many things? Three. And those three things are all branches of the contents of the porta hepatis, which are bile duct, proper hepatic artery, portal vein. And we see those branches on each corner there of the liver lobule. So at each corner you have a branch of the bile duct, you have a branch of the portal vein, and a branch of proper hepatic artery. So, any one of that set of three is considered portal triad. Now, I don't want to draw all the hepatocytes, but what you can see is that there are these channels flowing to the center called the central vein here. And those channels are blood vessels called sinusoids, hepatic sinusoids, all flowing toward the middle. And sinusoids. What's flowing in those sinusoids is a mix of portal blood and oxygenated blood from the proper hepatic artery. So the oxygen-rich blood flows down toward the central vein, and so does the portal vein there. So sinusoidal blood blood is a mix of blood from portal vein and proper hepatic artery. It's about 75-25. About 75% is portal blood, and 25% is blood from proper hepatic artery. The blood, it's flowing from the corners inward. So, I mean, just thinking commonsensically, you got cells that live closer to the edge. You got cells that live closer to the shower drain. Which cells get more oxygen? Yeah, the ones closer to the edge, closer to the blood supply. And you can quadrant out the lobule. We're not going to go to that detail, but I do want to know at least this, that's the blood that's flowing through. And as it flows towards the shower drain, that's when the hepatocytes have time to extract all the nutrients from the food you ate, from the portal blood, and the oxygen from the oxygen-rich blood. But by the time it gets to the shower drain, you're done. It's on the way out. There's nothing the liver can do about it. Okay. So to look at that in more detail, to draw some cells in with the sinusoids, I did a close-up and I blew it up, and that's why it's all pixelated. But what you should notice is hepatocytes are arranged in rows, in two rows. Okay, rows of two. So the name of the liver cell are hepatocytes. They're cuboidal-looking cells. These are the cells that extract the nutrients from the food you eat. They also produce the bile. Okay, that's the main thing I focused on. Put that as a side note. We already talked about how the liver produces bile. But anyways, arranged in rows of two. So it's like, here's a hepatocyte. There's one row of 4 cells. I want to draw another row right next to it. I purposely do that little big gap in there. That structure, that little hole in the middle right there, is called the bile canaliculi. That's the first structure you secrete bile into, actually. And it's a gap between cells, basically. On the picture, it's these green structures. However, I don't like how they're illustrated. My understanding is that it's just a gap between like the cell membranes of the hepatocytes. It's not a duct. Here it appears to be a separate duct structure, but that's not my understanding of how it really is. So But anyways, you still have to know bile canaliculi. And notice that each row is exposed to sinusoidal blood on one side or the other. So let's choose this side to have sinusoidal blood passing by. Okay, so as blood passes by, again, it's blood from portal vein and mixed with the oxygen-rich blood from the hepatic artery. And there's a small gap between this surface of the cell membrane and the sinusoid. Anyways, the side that faces the blood flowing by is called the basolateral membrane, and then the side opposite is the apical membrane. So this side of the membrane, basolateral, and then the side opposite, apical. So it's the basolateral membrane that faces the flowing blood. There is a small gap between the blood and the cells, and that's called the space of Dissay. SSE little apostrophe. And as the blood flows by, well, what I want to make about this arrangement is every cell is exposed to flowing blood. No cell is landlocked. It all can, like, whether it's on the top or the bottom of this picture, every row of hepatocytes has access to the blood flowing by, so the liver is very efficient in doing its job. All right, so to move on from liver lobules, talk about what bile is. Bile itself is affected because of its bile salts. Now, bile salts, you don't have to know the structure, but they include things like cholic acid, chenodeoxycholic acid. But this, I want you to know. How bile salts work is that they're able to have two sides, one side that can interact with water, one side that can interact with the fat globule that's trying to emulsify. Emulsification is simply breaking a big fat globule down into smaller fat globules, small enough to be absorbed by the lacteal. Bile salts are two-sided. One side is hydrophilic, one side hydrophobic. Well, let me just kind of draw there. So my green balls represent the bile salts. And please see the structure there. So this is, well, as the slide says, hydrophilic, space water. Space is the watery lumen, I guess, in the digestive tract. And the hydrophobic side, face fat. So fat emulsification, you're able to surround the fat and break it into smaller and smaller fat globules. And this slide shows a better picture of that. If you look at the top, that large fat globule has been broken down into smaller fat globules, little fat droplets surrounded by the bile salts. And what you're doing is the bile salts, by breaking the fat down into smaller and smaller droplets, you're able to expose more triglycerides to lipase activity. Fat emulsification breaks fat globules into smaller fat droplets. So you expose more triglycerides to lipase activity. I'll abbreviate triglycerides TGs to lipase to the T. So imagine if you had a big, large, fat globule, and my letter E's represent triglycerides. For example, if you had a big fat globule, the lipase can only attack and digest the triglycerides in the outer region, but the triglycerides in the inner region would not be digested. So if you break them down into smaller fat globules, the hepatic lipase, the lipase activity in the gut can digest better. And also, you're breaking things down into small enough molecules to be absorbed in the lacteal. This is in the small intestine. This is the villi. Smaller fat molecules. Well, they taste good, don't they? Make the food taste good. But anyways, in terms of digestion, smaller fat molecules can be absorbed in the lacteals. Okay, I'll put villi in parentheses. I'm reminding you where they are. That's the structure in the small intestine we talked about before. Now, so true digestions of fat. Fat emulsification is not true chemical digestion. Okay, but it helps. It makes it better. And the chemical digestion, the lipase activity, like I talked about, here's lipase. That's the enzyme that breaks down fat. You break off the free fatty acids from this is the glycerol backbone there. So these little these structures here are the free fatty acid, and that's the molecule that can circulate and enter the muscle cell to burn for energy. So when you burn fat during your workout, it's these molecules. Exercise also increases the lipase activity too. You can burn fat better during the workout. Okay, so anyways, I should probably note that. Lipase activity. You're increasing the circulating levels of the free fatty acids. You get them in the circulation, the muscle can use it for energy. Increase circulating levels. Of free fatty acids. So we're going to move away from liver and talk about the rest of the small intestine. We did talk about duodenum already, but the ileum and jejunum make up the rest of the small intestine. Together they're something like 6 to 7 meters long. That's about 21 feet. The jejunum is approximately the first 2/5, and the ileum is the remainder. Now, it's kind of hard to tell where one begins and one ends, but the beginning of the small intestine is the duodenum. It's the first 10 inches. You should be able to identify that. And you should be able to tell where duodenum becomes jejunum. Then it kind of becomes, it's hard for me to tell exactly when jejunum becomes ileum. There are clues, but you don't kind of know them. But so I would tag way down here, so there's no doubt it's ileum. You are expected to identify duodenum, jejunum, and ileum. Like for example, on this picture here, they're all coiled together. You can't tell where one begins, one ends. But if you tag close to the beginning, it's pretty clear. And if you tag close to the end, well, you should be able to tell then jejunum from the ileum. Now, the thing I focus on in terms of small intestine is absorption. We talked about intestines in terms of their mesentery. In this picture, they're all in there, but in this picture, they cut the jejunum where it begins there, and the ileum where it ends there. And I told you this picture before. I used that analogy of the python snake in the cloth bag. And well, anyways, if I had you identify this mesentery, the answer is root of the mesentery. That's one you got to know. That holds these intestines in place. And so I'm going to focus on their modifications for absorption, because that's their main function in digestion, absorb the food you ate. It helps that you're a super long tube, right? You have a long time to do it. It's about an inch in diameter, approximately. But that's not the impressive part, that you're an inch in diameter. You're 21 feet long. That's the impressive part. Let me erase this. Small intestine. Whenever you use the term small intestine, it refers to duodenum, jejunum, ileum. Total about 21 feet in length, 1 inch diameter. And so that's a long bit of length to accomplish absorption. So what they do is, if there's any digestion left to be done, you finish it off. Finish off digestion. I'll show you a slide that has the brush border enzyme to accomplish that. And also accomplish absorption. I mean, there's other functions too. I mean, one minor one, that the ileum has malt, and that has an immune function. We talked about malt already. That's there too. Focus on the absorption. You really want to increase the surface area, and the surface modifications that you have to know are the plica, the villi, and the microvilli. So instead of like reading from the slide, it helps to look at pictures instead. Now this picture here unrolls a portion of all the grooves, the circular grooves you see, but you see some finer detail here. So I'm going to teach you how the intestine increases its surface area for absorption. Modifications to increase surface area for absorption. If I draw just a small part of that, I don't know if you can see that. I'm going to draw this right here. See that little red line there? That's supposed to represent the muscularis mucosa. And then what you see is that you can see how the mucosa is folded in such a way all around there. So with all that folding, the big fold, this whole thing sticking up, that you can see with the naked eyes is plica, the plica circularis. So, what you got to realize is if you're folding all the way to the muscularis mucosa level, deep to it would be submucosa. So what I'm drawing in blue here is the loose connective tissue of the submucosa. But because you fold it all the way up like that, the submucosa goes all the way up inside of here. So again, the blue represents the loose CT of the submucosa layer. So what we see here is that the plica is actually a folding of this all the way down to the submucosa. So all these little, I'm going to draw all these little like green loose connective tissues. I don't want to draw for the whole thing, but maybe we can later. What do you think it represents? It's loose connective tissue of what? It's deep to the epithelium. Abbreviations, LP. The lamina propria. So. So what we say is that these little folds, these little grooves, I'll point to these three. Those represent villi. So they're just a folding of the mucosa. Okay. But anyways, both levels, plica, villi, you're increasing the surface area. This big fold that you can see with the naked eye is increasing the surface area for absorption, and so are these little ones, the villi. And you even fold at the cellular level. So I have a slide showing you, I should have started with this. So you can see now close up. This is muscularis mucosa. I mean, deep to it is submucosa. So above it is like lamina propria. That's plica. So just looking at these little finger-like projections, I didn't draw the pits. I just drew the villi sticking up. That's a villi, so that's a folding of the mucosa. Now, if you look at the epithelial cells lining, say, an individual villi, each individual cell has a brush border microvilli. So that's kind of the third level. Kind of mentioned that before. This is a columnar epithelium. Cell of small intestine. What we'll see is that the folded end are microvilli, but it's going to be studded with brush border enzymes to finish off the digestion, as well as absorption. So I'll just draw a few little dots there to symbolize brush border enzymes. That's this slide here. If you have any polymers that need further digestion, they can finish it off, and then on the other side of the cell, you can absorb the monomers into the bloodstream. So here's a model of the small intestine that we looked at before. It has all the layers. It has MALT. It has the villi. It has the crypts. You can see the individual epithelial cells along with some goblet cells there, and it's labeled on the other side so you can quiz yourself. And I also taught you this one before too, so both of those are there. And the model's in the room, so you can study it. And so once you accomplish absorption of the small intestine, that absorbs most everything. And by the time you're make it through that 21 feet of intestine. Now you're into the colon. The colon, or large intestine, I use both terms. When it's isolated, it looks like this. Okay? It has different parts. So we're moving away from small intestine to large intestine. Now the large intestine is not longer in length, but the diameter is greater. And that's about 2 to 3 inches in diameter, whereas the small intestine is only about 1 inch in diameter. What I'll do is when I get the cadavers out on Friday, I'll put them side by side so you can see the girth. Let's just list all the parts of the small intestine, or excuse me, large intestine, or colon. Large intestine. Includes the vermiform appendix, this is the worm-like structure that, if it gets clogged with fecal matter, can strangulate the blood flow, and it has to be removed or it bursts. Right there. Okay. Right there. This little pouch, which receives the food stuff from the ileum, that's the cecum. For it to receive food stuff, it needs an inlet. So you see those little lips right there? They say ileocecal labrum, superior, inferior lips. I usually just call this the ileocecal valve right there. So within the cecum, look for ileocecal valve. So vermiform appendix, cecum. One thing to note, and it's kind of hard to tell because they cut away the front view here, so you can see the little lips of the ileocecal valve is. At the cecum, the longitudinal layer of muscularis externa, it narrows into 3 bands that are longitudinal. So the longitudinal layer of muscularis externa is not a complete layer in this region of the GI tract. It's reduced to 3 bands called the tenei coli. Look for those. Right there. 3 bands. Okay, but what they are in terms of the GI tract wall, longitudinal layer of ME, muscularis externa. Longitudinal layer of muscularis externa, ME. So you can kind of see them. You can see some of them on this picture. We went up along the side there. Well, anyways, there's tenei coli there. And so because it's kind of like a drawstring. So when you cinch a drawstring, it kind of creates all these little pockets when it contracts. Those pockets have a name. They're called hostra. Pocketed appearance of the large intestine. That's one way you can tell large from small. The small intestine is smooth, but because of the longitudinal layer is reduced to 3 bands, like it creates these hostra. So that's another thing I'll have you identify. Okay, so there's all those terms. To continue with the includes ascending colon. we just left the small intestine. The small intestines are intraperitoneal. It kind of goes retro here. Retroperitoneal. You can see it's about 12 to 20 centimeters. It ascends retroperitoneally up the right abdominal cavity from the cecum. I said it does contain the vermiform appendix. So in anatomy, use the full term, vermiform appendix. Vermiform means shaped like a worm, okay? Well, that's misspelled. Let me put an M in there. It's spelled right in here. So I'm going to fix that later. This is vermiform. Vermiform means worm-like in appearance. Well, then you kind of make a sharp turn here. You ascend up, and that right turn is called the right colic flexure. It's also called hepatic flexure because that's by the liver. Always remember, liver on the right, spleen on the left, liver on the right, spleen on the left, liver on the right, spleen on the left. I always recite that to myself, so I never forget. Hepatic flexure. Okay, so you make that turn, then you're in the transverse colon. Notice I'm kind of like, it's not straight across, it kind of like loops down. It's following the curvature, with the greater curvature of the stomach, basically. It kind of loops down. If you ever have cramps, it could be your stomach, it could be the transverse colon. They're kind of really close to each other. So from right colic flexure, transverse colon. Now you go back intraperitoneal. You were retro, now you're intra. So you go from the right colic flexure, about 45 centimeters across, to the left colic flexure on the other side. Also called splenic flexure. Because the spleen's on the left. And now you're going down. And the descending colon goes back retroperitoneal. And it stays that way until the end. Descending colon. Goes back retro. The descending colon is about the last, well, it's not the end, but it's about 22 to 30 centimeters. It descends, it's retroperitoneal from the left colic flexure on down. Then where it takes kind of an S-shaped turn, it's called the sigmoid colon. S-shape. So it joins the rectum, and what you can see at the rectum is you kind of lose the pockets because the tinea coli, you're reforming the full layer of the longitudinal layer of muscularis externa there. So that's why you lose the hostra and you go back to just a regular full layer of that longitudinal part. So I'll say at sigmoid rectal juncture, you reform the full longitudinal layer of the ME, muscularis externa. there are some pictures there, and you can see the small intestine green, large intestine gray. You can see part of it there. If I point to like 3 little pouches or something, I'm going for a hostra. If I point to this like whole thing, what do you think I'm going for? Transverse colon. What if I just put like a little line just on that part, that little ribbon? That's that little Tenei coli thing. Tenei coli is hard to spell, and it's spelled many different ways. That's one where I'm really lenient on the spelling. Try to come close. Okay. One time I looked at it in like 4 different books, and they all spelled it differently. Okay, well anyways, more of the intestines here, and okay, it's like 527, they're going for the hostra. Let's see, yeah, it's pretty straightforward. Let's see here. What do you think 528 is? Yeah, I'll put descending colon. 526 is the Tenei coli. Remember 491? Duodenum. 496, pancreatic, the main pancreatic duct, the pancreatic duct. 495 is the pancreas, obviously. Okay, let's get to the end here. So when you get to the rectum is 12 to 15 centimeters. It's the fixed terminal part. They say it's fixed because it's following the curvature of the sacrum. It's kind of attached to there. It's continuous with the sigmoid colon. Rectum. Well, in case you didn't know, this is where feces is allowed to accumulate. Accumulate. How about collect? Now, so what has happened is, all along the large intestine, you're pulling out water and fat-soluble vitamins, and that compacts the foodstuff into fecal matter. Absorption of water, which occurs in the large intestine, compacts foodstuff into fecal matter. So in the rectum, when the feces, it distends, stretches, rectal walls, they contract. I think I mentioned this before. You push toward the anal canal. They do show that there. What I want to focus on are the sphincters. This is the anal canal, bounded by the external, the internal anal sphincter. The external sphincter is that's skeletal muscle, that's voluntary muscle. Push toward anal canal. And that's when you feel the urge to go. And it's the external sphincter you can control with conscious thought to hold it in until you get to the bathroom. External anal sphincter. Yeah, so I'll just note that that's it's a voluntary muscle. Okay, Okay, skeletal muscle. So that's the end of the digestive tract. So I do want to talk about, shift gears and talk about the blood supply to this area. And I've already mentioned the superior mesenteric artery, and that's shown here. What you can see is branches of the superior mesenteric artery. I mean, you don't have to know the names, just know that ultimately all of those branches shown here, they're supplying blood to the jejunum, also to the colon, all the way up to here and here, okay? Here is showing you the complete blood supply a little bit better, and I have a model too that shows it there. That model is in the back over there, you want to look for it. Well, anyways, here's what I would note. It supplies jejunum ileum as well as colon, all the way up to the left colic flexure. Or splenic flexure. And that's what's shown on the left there. Notice how it kind of goes off to the right. Okay, that's kind of consistent with cadavers. It goes off to the right, and as it goes off to the right, all these little branches coming off of it are giving a blood supply to the jejunum and the ileum. On the model here, here's how they show it. Here's the superior mesenteric artery, and all of these branches are going to go off and supply jejunum ileum. And then it's also supplying blood all the way up to there. Now they show it as a continuous thing, but the blood supply to the colon, it's ending there. That's the left colic flexure. So just to orient you with this picture, if I'm telling you that's left colic flexure, what flexure is that? The right one. So what's this structure here? Ascending colon. So it's going all the way up there. Just make sure you see that right correctly. So here's the small intestines properly placed inside the large intestine model. Now I wanted to show you the backside of this. All those little cut branches, the red and the blue, are superior mesenteric artery and vein there. So all of these little cut branches, they match up with these branches here. Okay, so when you kind of put this into there, they're fitted together. Okay, so that's how it's modeled for you. That's the superior mesenteric artery. So the rest of the distal colon is supplied by the inferior mesenteric artery, shown here. It supplies from left colic flexure down to the end. I'll just say on down to the end. Now, the superior mesenteric kind of goes off to the right. The inferior mesenteric, it kind of goes off the other way, to the left. There it is cut. There it is not cut. You can see the branches from the distal colon on down there. Okay, that's what it supplies. It's the inferior mesenteric. And they're not hard to tell apart. I mean, if you could see them both, the one on top is the superior. It's the one that's going to go over the duodenum. I mean, just look. If you get close to the branch point there, that's the inferior one. It's a single. It's not paired. one to identify on cadavers. I see it quite easily. All right, so anyways, up to the left colic flexure, that's SMA, and then inferior mesenteric just gets the rest of it on down. And then some more pictures. But that's the superior one, the one next to it. And look where all the branches go. That's inferior mesenteric. Okay. Here they cut it there, but they didn't cut the colic branches of the superior mesenteric. And you can kind of see how the inferior mesenteric kind of picks up right there and goes on down. More pictures. Once again, if you know it's going over the duodenum, that's the superior one, making that the inferior one, even though you can't fully see it. But there are enough clues so you can identify it more easily. So if that's the blood supply, the drainage is by the portal vein. And you should know that too, because portal circulation goes to the liver first, and we call that first pass, because all the blood must first pass through the liver, shown here. So notice how there's no inferior vena cava. All of these structures flow to the liver first. The single drainage system of the liver are the hepatic veins, not shown here. But notice how portal vein is a blood supply. Okay. All right, so in portal circulation, you're draining all the blood from the intestinal capillaries, and the portal blood goes right to the liver sinusoids that we talked about earlier. So I think what you should know from this slide, let me cut to some nice pictures. I think the one that's most useful is this one here. So I'm going to start with bottom up. You got the blood draining from superior mesenteric vein, inferior mesenteric vein, splenic vein. They're all going to merge together here. See, so superior mesenteric vein. Inferior mesenteric vein. Then you're going to get splenic vein. From spleen. They merge together. Portal vein. And this is going right to liver. So this is portal circulation. And all the blood is flowing up to portal vein. So those are the veins I would study for the portal circulation. And that kind of concludes, the gross anatomy. I do want to talk about the histology now. I do have some pictures from the histology on Canvas. The ones here on this slide, I don't teach from them anymore because I've taken other pictures since then. When I first made these slides, but I just left them there because they're still good to look at. But I'm going to go straight to the ones I posted yesterday, last night on Canvas. And this will help you get ready for the histology quiz. The histology quiz is listed on the schedule for Wednesday. It's going to be on Proctorio, but I'm going to make it available today through Wednesday. So you can do it whenever you want, today, tomorrow, or Wednesday. It's on the schedule for Wednesday. So I'm going to make a modification to my camera here.